Welcome back to Menopause Mastery, lovely ladies. I'm your host, Dr. Betty Murray, and today we are going to take a deep dive into what I think is a very crucial bit of science for women, and it's all about how our hormones are metabolized, and particularly a connection to breast cancer and ovarian cancer risk, and a little bit about my hypothesis. So many of you, if you've listened to my show before, know that this was part of my actual dissertation for my PhD. So I am heavily immersed in how our body takes estrogen and your hormones, other hormones like progesterone and testosterone, and goes through the complex process of wrapping them up and getting them ready for the trash. So I'm going to go into it today, but I want to go into a little bit of this because I want to share a little bit of my hypothesis. So first off, scientists have hypotheses. Hypotheses are their guesses. Is this true or is this not true? And when you go to do scientific research, often it always starts with a hypothesis. You have an idea of something that you think may be causing something else. And your job is to, number one, go out there and look and see if anybody else has studied it before or studied something like it to sort of understand the available data out there and what really already has been done and what may need to be modified or improved. You know, it's a big part of the entire dissertation process and the scientific process is to look what's already been said and then to actually dig through the data. So not just read the abstract and regurgitate what you think they said, it's actually digging through the actual research. Number two, it's also from an academic and scientific integrity standpoint, your job is to go out there and really then try and disprove it, right? So if you go to prove your hypotheses, you automatically have bias. You know, you're automatically sort of selecting things that follow your mindset. And we have a lot of bias, and it's impossible to get rid of all of it. But in scientific exploration, the most important thing you can do as a scientist is try and, number one, limit your bias, and you must call it out. Right? You got to say, this is some things where bias could show up. And then your job is to create a hypothesis of it being true or not true, known as null. And then your job is to go out and run your scientific experiment and try and disprove your hypothesis. So gather information that would make it not so, because then you are actually doing the right thing. So we're going to talk about some heavy stuff today, and specifically around estrogen metabolism, because I still, I do these weekly webinars with women about hormones and hormone replacement and the safety of hormone replacement. And so many women are terrified of hormones because they think it's going to increase their risk, particularly for breast cancer. And, it's because the Women's Health Study, and actually the women in that study that were on estrogen only had a 23% decreased risk of breast cancer. But of course, that wasn't really popular. That wasn't a popular statement because we didn't want to promote that. We wanted to promote fear. But when you get right down to it, there is this increased risk for estrogen-sensitive cancers post-menopausally, right? Whether it's ovarian cancer, uterine cancer, or breast cancer. We have this increased risk at the time when our hormones are absolutely at their lowest. Your ovaries have petered out. There's no eggs left in the carton. It is done. So it defies logic. And so again, I've spent a lot of time in the research, and what I want to share with you today is sort of my working hypothesis and some of the things that I would love to help you understand so you could understand why I'm so passionate, number one, about I think hormone replacement's on the table for pretty much all women. And I think, our paradigm, particularly that we do clinically and then also that we are doing with our telemedicine company, is so very valuable because we're going way further than what conventional medicine looks at, because we're actually looking at the pathways I'm going to talk about today, which is, I think, Peter Attia likes to talk about a medicine 3.0. This is medicine 3.0 on steroids. And it's because we need to use more data to make decisions. So grab your favorite beverage, get comfy, because we're going to talk about estrogen today. So let's talk about estrogen first. Let's start with the basics. Estrogen is kind of like a Swiss Army knife of hormones in your body. So it's not just one hormone. Estrogen, actually, the hormones that we make in our body, we make three forms of estrogen. We make estradiol, estrone, and estriol. And they play various roles. They do things like regulating our menstrual cycles, to maintaining bone health, to remodeling the arteries, to protecting the heart, to helping your powerhouses inside your cell utilize glucose better, brain cells being able to create memories and focus. they are involved in every type of interaction in your body. And actually, every single cell in your body, except for two, has receptors for estrogens. So basically, there's a keyhole on every single cell, except for red blood cells and a single type of cell in the brain, only one out of the many, that don't have a keyhole for estrogen. So that's why we get so many symptoms. It's so varied, and it really depends how your body is expressing those receptors and whether they're getting fed or not. Right, so here's where it gets interesting. So our body doesn't just use estrogen and kind of wave it bye-bye and say, okay, yeah, just bye-bye, let it go. We actually have this really complex system of what's called metabolizing estrogen. And this happens through different pathways, and it happens in the liver. And these different pathways involve different genes and different enzymes. So think of it as multiple exit routes, and we have multiple ways to get things through that exit route. And ultimately, how you're wired genetically influences sort of which pathway you go down. So the first one is called hydroxylation. So I'm going to use these words. I don't expect you to remember them at all. So we're going to use a concept I like to call prepping for the trash. So when your estrogen is ready to get thrown out, and this is true of all your molecules, testosterone goes through this, progesterone goes through this, your body utilizes it. It's kind of like a highlighter or a marker that's sort of drying out. It doesn't work very well anymore. You've already kind of used it up. So you need to throw away the carcass of the highlighter, right? The marker part itself. So you can't just take the marker and throw it in the trash, right? So you can't just take your estrogen and throw it in the trash. You've got to take your estrogen and you have to wrap it in these different colored wrappers to get it ready for the trash. The color of the wrapper is distinguished by the step or the step in the liver process of sort of prepping it for the trash. And it's also determined by your genes. So for instance, the first step is called hydroxylation. You don't have to remember that. There's no tests at the end of the podcast for this. Hydroxylation is where your body's adding what they call an OH group, right, to the estrogen. So it's not breaking something off the estrogen, it's actually wrapping it on it. And that process happens at the very first step, or what we call phase 1 of liver detoxification. And this is where the toxin that is fat soluble is getting converted. And at hydroxylation, when they stick the hydroxyl group on there, right, the OH on there, it actually makes it more reactive. So that's the first thing. So when the liver takes and gets ready to do that very first process, the thing that is made intermediately is actually more toxic, right? So it's more damaging, right? And it's got varying effects on our body. So that very first step, we have 3 genes that control it. CYP1B1, again, you don't have to remember these, they're called CYP1B1, CYP3A4, and CYP3A1, right? So you don't have to remember those. So here's how to think about it. This is my color-coded tissue paper description, which I think is a great description. So we have one hydroxylation pathway that makes a 2-hydroxy estrogen metabolite. So the 2-hydroxy is a green wrapper. So imagine I've got a bunch of different tissue paper wrappers. I'm going to wrap up my dirty, funky highlighter that needs to get thrown away. And I've got a green tissue paper wrapper that I can wrap it in. That's made by the 2-hydroxy pathway. That is considered the most clean pathway. That is considered the healthiest pathway, the best pathway. We want to have genetics for that pathway to do a lot of that. I also have another pathway ran by CYP3A4, which has a blue wrapper around it. That one is less favorable, but it's also the way our body makes another estrogen called estriol, but it's less favorable. So we want some to go through that pathway, but not a lot. And then I have one that wraps a red wrapper around it. That's CYP1B1, right? The red wrapper, I don't want to make a lot of that. That one makes a 4-hydroxy estradiol or estrone, which is a metabolite, right? So it's that intermediate byproduct. That one is considered toxic. It is DNA damaging. So if it's circulating around and banging around on cells, it can actually damage the DNA, which is how things like cancer cells can proliferate. And so we don't want a lot of the red wrapper estrogen to get stuck, right? So that first step in the liver, we either wrap a green, a blue, or a red wrapper around it, right? We want a lot of green, very little red, and a little bit of blue, right? Now, the next step, we stick a methyl group on it. It's controlled by methylation. A lot of people know about methylation because they've read about MTHFR and methylfolate and that gene, but there's other genes that are involved in methylation, right? And this is This is adding a group around it. So I like to call this the pink wrapper, and it's controlled by a gene called COMT, C-O-M-T. And so if I have plenty of methylfolate and plenty of methylcobalamin, which are the methylated forms of B9 and B12 respectively, so if I have plenty of folate, plenty of B12, my COMT gene is either really good at wrapping pink wrappers around those either green, blue, or red wrappers, or they aren't, right? Or they aren't. So if I am deficient in those nutrients like folate and B12, I won't have any pink wrappers. Right? So that means the green wrappered stuff or the red wrappered stuff or the blue wrappered stuff just circulates. You can't get rid of it because it's got to go through the pink wrapper. It's got to stack, right? I have to have multiple colors on there. So that methylation process puts a pink wrapper on it. Now, COMT, you can either be a fast COMT where you make a lot of pink wrappers, or you could be a slow COMT. Like I happen to be a very slow COMT. I don't make a lot of pink wrappers. So I have to be very mindful of the fact that I'm not very good at that next step. Well, research shows that if you have estrogen metabolite stuck where the green wrapper's on it, the red wrapper or the blue wrapper, and it can't get to the pink wrapper, and there's not enough pink, we got bad estrogen metabolites and we've got an increased risk for cancer, right? So we have to have both of those go in order, right? And I still want more green wrappers than I do red, and then I want a lot of pink wrappers to wrap around all of them. Right, so that's the methylation process. That's phase 2 of liver detoxification. Let me call it, 1.85. It's kind of into. So now that I've got my estrogen wrapped either in a green, red, or blue wrapper, and then an extra pink wrapper around it, I have 2 ways it can exit. One is called sulfation, and it's where we add a sulfate group to estrogen, making it more water soluble. So this has to happen. It has to become water soluble because otherwise it becomes what we call fat soluble, and it'll stick around because it can't get excreted. So sulfation it takes that pink wrappered estrogen metabolite and wraps an orange wrapper around it. I need to have a lot of sulfate groups there supporting it. Where do I get sulfate from? So I get it from foods that are high in sulfur, right? I get it from proteins that get broken down as long as they make it all the way to the sulfate form, which is another conversation. I get this from my diet, right? And so I've got my sulfate that wraps around the estrogen, puts an orange wrapper around it. That gets it to the dumpster. I have a secondary pathway called glucuronidation. This pathway adds a thing called glucuronic acid to the estrogen. So think of this as a black wrapper. So it's a secondary pathway where the estrogen actually gets excreted through the stool rather than the urine. Sulfation, that orange wrapper, that gets extracted and sent to the urine. That's the preferred pathway for estradiol, which is what your ovaries make. I'm sorry, pardon me. The sulfation pathway is your preferred pathway for estrone, which is a secondary estrogen, which is more pro-inflammatory. Your estradiol pathway, and then estradiol goes through glucuronidation more heavily, and it goes out through the stool, right? Both of them make it water soluble. So essentially, here's the take-home message here. I know it's pretty heady stuff, but I know you guys are brilliant and you can get this. In order for your body to excrete estrogen and anything that looks like estrogen, pesticides, herbicides, plasticizers, BPA, all that other stuff, birth control pills, you name it, the body has to go through each one of those steps. It's got to wrap it in a red, a green, or a blue wrapper, and we want a lot of green. It's got to wrap it then in a pink wrapper. I need a lot of pink wrappers because I got to wrap up all the ones that were made. And then I've either got to put it in an orange wrapper and take it out through the urine through sulfation, or I've got to put it in the black wrapper and it's got to go out through the stool. Why this is important is it's not so much whether you have one or more of these, it is the relationship between that, and that seems to play a role in estrogen-sensitive cancers. And it's more about not just having the genetics, but it's more so about what are these in combination to each other. And how do they relate to each other? So the breast cancer risk, particularly, which is where a lot of the research, but even endometrial cancer, has shown that the way our body metabolizes estrogen, right, how we get it through that process of the different colored wrappers is not just about how much estrogen we have, but how our body deals with it, how it gets it out. So for instance, if I make a lot of the green wrappers and I get a lot of estrogen that way, which makes 2-hydroxyestrone. It's generally considered protective against breast cancer. So I want a ton going through there. Those metabolites, the green wrappered estrogen, is weak. It doesn't bind heavily. But if it goes on down the 4-hydroxy pathway, which is that red wrapper, it has been shown to increase breast cancer risk. And same thing with the 16-hydroxyestrone. So the blue and the red wrappers have a greater risk because they have a stronger potential to grab a hold of an estrogen receptor on a cell and exert activity, potentially damaging the DNA. Because you have to remember, these aren't real estrogens like what your ovaries make. These are sort of halfway, half damaged. And so it's not just about the individual pathway, but it's about a balance between them. I'm going to talk about a study that was concluded in 2020 by Trabert and colleagues that looked at the estrogen ratios and how they played out and the reason why this is so important. So, for instance, they were looking specifically at endometrial cancers, and what they found is women with higher levels of estradiol, so that's the one your ovaries make, in their blood, were 4 times more likely to develop cancer in the lining of their endometrial lining when unopposed with progesterone, right? Progesterone and estrogen play together. It's kind of like you have to have, both sides. Just like yin and yang, you need both together. If you give estrogen alone without, or you've got a lot of estrogen without adequate progesterone, you're going to have problems with cellular proliferation in the uterine lining, so cells building up and especially blood supply going in there, which are both things that you don't want to do. However, when they were looking at it, they looked at how the body breaks down these estrogen, these metabolites, right? The things I was just going through. And they found that women whose bodies produced more of the breakdown product, the 4-hydroxyestrone, or otherwise known as catechol estrogens, and the 16-hydroxyestrone, compared to or relative to the methylated estrogens, right? So the pink wrappered stuff were at a higher risk of endometrial cancer. So ultimately, it was those liver byproducts that weren't properly prepared for the trash that increased that risk. And women with the highest levels of 4-hydroxyestrone and 16-hydroxyestrone, red wrapper or blue wrapper, were 4 to 4.5 times more likely to develop endometrial cancer compared to those with the absolute lowest levels. Right? So what does that tell us? The key finding shows that both higher ratios of the 4 catechol estrogens and the 16 were associated with increased risk of endometrial cancer, independent of estradiol levels, right? So why is this important? I started the conversation early on pointing out the fact that endometrial cancer and breast cancer is higher risk when we don't have estradiol anymore. In a menopausal woman, estradiol levels are almost non-existent. That means your ovaries are out of eggs. There's none of it circulating. However, the standard of care is if someone has an estrogen-sensitive breast cancer, is we shut down any production of estrogen. But we still have estrogen metabolites going through the liver. We still do. Because our fat cells make estrogen, the toxins in our environment make estrogen, and what Trabert showed was that those byproducts, if not properly prepared for the trash, were independently a risk factor for endometrial cancer, right? So sort of put that in this little bucket and let it sit there for a minute because I'm going to circle back to it, right? So the take-home message, very simplified take-home message, is when those metabolites that are the first step, phase 1 liver detoxification, where your body puts a blue, a red, or a green wrapper around that estrogen, if it can't get the pink wrapper on it, we got a greater risk. Right? We've got a greater risk, regardless of whether you've got estrogen on board because you're on hormone replacement or not. The risk is present, right? And I'm going to come back to some thoughts that I have about this and my working hypothesis, right? And it was significant, right? So these ratios that compared these two byproducts, basically that stage 1 of liver detoxification or stage 2, where the methyl group, the pink wrapper, gets put on it, is where the risk profile really became higher, right? What they also did, which I think is important, so again, I even put this out on social media, women are always like, I don't want to take estrogen replacement because I'm afraid I'm going to get breast cancer. Well, obesity increases your risk for breast cancer, depending on the study, anywhere from 21 to 32%, right? Independent of everything else. So if I am overweight, I am almost 30% more likely, higher risk, more likely is not the right way of saying it, but higher risk to get breast cancer. even at any crazy estimation off of the Women's Health Initiative, which absolutely did not show that, it wasn't that high of a percentage, right? In that arm that they misspoke when it went from 4 in 1,000 to 5 in 1,000, which yes was a 25% increase from 4 to 5, but it was not statistically significant. That's nowhere close to the risk when we have excess body fat that we don't want. Because again, what does fat do? Fat makes an estrogen that is not estradiol. It makes estrone. It makes estrone, which our body has, but it shouldn't have a lot of. We need to have estradiol. Okay, so BMI increases your risk. So when they controlled in this study for BMI, it still suggested that this ratio between the liver detox pathways are significant risks. So they even controlled for body weight, right? And other risk factors, right? So interestingly enough, when they looked at ovarian cancer, and that wasn't the focus of their study, but they sort of stepped off and looked at ovarian cancer, they didn't find any clear connections between estrogen-related substances and ovarian cancer. So in simple terms, the study suggests that it's not just about the estrogen being in the body, but it's a matter of how the body processes and prepares that estrogen for exit, right? Getting it to the dumpster. And if the body isn't very good at that, that's where we have to pay attention, right? So this information really, I think, is valuable. I know it's a heady thing, and you might have to go back and listen to it again. But I want women to understand it's not so much about the hormones that your body makes, because if it was, we would all have cancer at 20 when our hormones are wildly high and wildly variable, right? Or pregnant, right? Your estrogen levels can climb to 4,300 when you're pregnant. When we replace hormones, I don't because I'm not a prescriber, but when prescribers replace hormones in a menopausal woman, you're shooting for like a 60 to 80 on labs, not 4,300. It's 1/10 of what is your natural range under a normal cycling time, and significantly lower, even more so than what we put young girls on without ever questioning it when we put people on birth control. So it's very, very low. It is about how your body processes estrogen-like compounds. And especially when we look at all the toxins in our world, that can have an effect, right? So what this also probably brings up for a lot of people is, well, what if I have genes in those pathways that make me make more red wrapper or more blue wrapper compared to green? Well, if you've ever listened to my show, you can go back to some of the early shows where I talked to Kashif Khan, and I actually share my personal genetics online. He and I are talking directly about my genes, and I happen to have heavy, heavy mutations in this pathway, which is why I know so much about it, because I went back and researched it because I wanted to understand it. And the beautiful thing is you can't change your genes, but while we can't do that, we can influence how they're expressed. So quite literally, how you eat, the supplements you take, the lifestyle you have helps you make more green wrappers, or if you live a really bad lifestyle, makes more red wrappers. So we can absolutely influence this, and especially if somebody's doing hormone replacement. We can check it, we can monitor it, and we can modify it, because these are epigenetically modified genes. Epigenetics are what you eat, think, do, breathe, come in contact with, take. And epi means on top of the gene, turning it on, turning it off. So that's where it gets exciting, right? So now I want to talk a little bit about the lifestyle factors and how they play a practical role. So again, if I have less than favorable genes, I want to support estrogen metabolism. And here's the thing, even if you don't know your genetics, right? We do genetic testing when we're working with people. We also check and make sure we know how you are getting through these pathways. We have urine tests that we use to do that. And these things can be checked. I think that's the first thing you need to know. Now, conventional physicians are not going to do that. They don't even know how to read this stuff because they don't get trained in menopause training, and they definitely don't go back and learn hormone metabolism. You have to find a functional person or work with somebody like on our team to get there. But you can absolutely look at this. So let's talk about, obviously you can test for it, you can find it, you can see it, we can monitor it. Like I check my hormone pathways once a year. I want to know that my exit routes and my dumpsters are getting all the way to the street every single time, whether I'm on hormones or not. Because if I'm not on hormones, my risks go up anyway, right? For some sort of cancer, particularly an estrogen-sensitive cancer. All right, so let's talk about what does influence these hormone pathways, right? So what positively helps you make more happy green wrappered estrogens and pink wrappered estrogens, and you get them all the way to the orange, wrapped dumpster or the black wrapped dumpster. So a review in 2021 found that regular exercise, right? So significant regular exercise. So, walking every day, adding weight training. They did look at moderate aerobic exercise. It's easier to study than weight-bearing exercise. It was associated with a 25 to 30% reduced in average risk in breast cancer in women, right? So let me say that again. Exercising consistently and regularly will reduce your risk of breast cancer by 25 to 30%, right? This is Soljenska and colleagues in 2021. So this study suggests that moderate aerobic exercise, obviously combined with a good diet, is going to reduce our exposure to these estrogen metabolites and help your body get the things out of the liver and get them to the dumpster. So exercise not only makes you feel better, makes you maybe lose some weight or gain more muscle and all those other things, it actually helps your body detoxify by helping those pathways. Now, diet, what you eat, has a significant influence on these pathways. And I'm sorry if any of you are carnivore, but I got to break it to you. Carnivore is not going to do positive things to your hormone pathways. We need vegetables like broccoli, cauliflower, sulforaphane. That's the ingredient in there, and indole-3-carbinol, and the sulfur vegetables, onions, garlic, all of those things. And the reason why is they positively push that estrogen pathway to make more of the green wrappered estrogen metabolite. So it helps you make cleaner estrogen metabolites. Also, things like flaxseed, cruciferous vegetables, berries also improve that. Hops, citrus bioflavonoids, so having citrus curcumin, also positively affect these pathways. It helps push things out of that red pathway, the 4-hydroxy pathway, into the other green pathway. Even things like green tea catechins, right? So lots of vegetable and plant-based foods have positive impact on hormone metabolism. And directly, we're going to talk a little more about what else they do. And so these foods, particularly the Brassica family foods, have been shown to support these pathways over and over and over again. That's why broccoli is associated with lowered cancer risk. It's because it literally affects these pathways. Phytoestrogens. So phytoestrogens are plant foods that have wheat-based plant estrogens in it. So for instance, soy genistein, flaxseed, all have phytoestrogens in it. And they actually have been shown in studies that they actually have a positive protective effect against cancers, estrogen-sensitive cancers. A matter of fact, a study done by Sweeney and colleagues found that phytoestrogens, particularly genistein, found in organic non-GMO soy and equal, can have complex effects on breast cancer cells. At a low concentration, if it's not very much, could be stimulatory, but higher concentration, it actually inhibits death and even induced cell deaths in some breast cancer tissues, right? So it doesn't mean you need to go down a bunch of soy milk by the gallon, but it does mean that some of the phytoestrogens, my preferred phytoestrogens, are things like your flax meal and flaxseed. Even sweet potatoes have some phytoestrogens. Red clover has some phytoestrogenic activity. Those things are very, very helpful, right? That moderate consumption can actually be really, really helpful. Now, resveratrol, right? So this is found in grapes, right? And yes, it's found in wine, but I hate to break it to you. You'd have to have like 30 bottles of wine to get any kind of reasonable amount of resveratrol. Do not fall for the alcohol conversation, which I'm just going to make a quick sidebar and talk about risk for breast cancer and ovarian cancer. If you drink one drink a day, your risk for breast cancer is 7 to 12% higher. For every additional drink, it goes up 7 to 12. So if you are a daily 1 or 2 glass or more wine drinker, or whatever it doesn't matter what kind of alcohol, your risk for cancer goes up, right? It is absolutely damaging to all cells. Do not fall for the fact that wine has resveratrol and it's protective. That's just marketing, right? I like a really great glass of wine just like everybody else, but you have to recognize there is no health benefit. And if you have a lot of concerns around breast cancer, this is a place to make some serious action. All right, so let me go back to resveratrol, right? So one study found that resveratrol can actually alter estrogen metabolism in breast cancer cells, so petri dish, change how estrogen is metabolized. And again, this is another thing is if you have small amounts, it actually might stimulate cell growth. But if you take large amount of resveratrol, like you might do in a supplemental form, it inhibits growth. And this doesn't mean that you need to go out and take a bunch of resveratrol. It's just important to understand that plant compounds and eating a wide variety of plant compounds from a wide variety of plants actually have significant effects on our hormone pathways and particularly how our body prepares things for detoxification. So we need them, right? So if you're not eating vegetables and fruits, you're not getting those foods. You can't get that. You cannot get those. You can't get sulforaphane out of a piece of beef or chicken. It just doesn't exist, right? I eat meat. I just recognize that it can't be the only thing that I eat. Now, NRF2. So NRF2, I'm not going to go into. It's a protein. It protects cells, right? And NRF2 has been shown to protect estrogen-induced cancer cell growth, right? So what we want to do is activate NRF2. A review done by Guidice and colleagues in 2019, this was a paper I dug in pretty heavily on during my dissertation, found that phytochemicals like resveratrol, sulforaphane, can activate the NRF2 pathway, providing extra protection against DNA. So this steps out of the hormone pathway and actually protects the DNA inside the cell itself from being hijacked as a cancer cell, right? So it keeps breast cancer cell initiation from starting. Obviously not a single bullet, you can't just take a supplement and it's going to be magic, but a part of a varied healthy diet and appropriate supplementation, especially once you know your hormone pathways, can absolutely make a difference on the risk factors, right? So we got to eat these foods. Now, one of the things that's really important, and it's something that we don't understand enough about yet, this was the second focus of my dissertation. It was the microbiome. Right? So our microbiome has obviously been getting a lot of interest in the scientific community, and that's because we have better ways of actually identifying those microbes and what they're up to. Metagenomics is the most cutting edge, which is looking not only at what bacteria and viruses and phages and archaea and all the little beings that are in our gut, which ones are they, but it's also able to look at what do they produce, what enzymes do they produce, proteins do they produce, and the whole collective DNA of those microbes. So you are not just your DNA that decided, what you look like and all those other things. You are the collective of your microbiome's DNA and what they produce. So we have a microbial community called the estrobolome, so estrogen bolome. Right? So these are bacteria that make a enzyme called beta-glucuronidase that is made in our body. We make it too, right? But beta-glucuronidase in the gut takes that estrogen metabolite. So remember the estrogen metabolite that goes through and gets the black wrapper on it at the very last step, and it goes out through the stool. Well, when you have a lot of bacteria making beta-glucuronidase, what it does is it pulls that black wrapper off of that estrogen-like compound, even if it's a pesticide, even if it's an herbicide, and it allows that compound to get recirculated back into the bloodstream through the liver, right? It goes out through the liver and back into the bloodstream, called hepatic recirculation. And that level has been associated with breast cancer, colon cancer, chemotherapy, drug-induced toxicity. So if I have a lot of microbes that are making beta-glucuronidase, I'm going to have a greater likelihood of my estrogen metabolite that's almost to the exit door getting thrown back into the bloodstream. So to give you a graphic of that, you've got the dumpster in the garage, and it's full of all the black wrapped trash, and somebody opens up the garage door and you throw it back into the kitchen all over the floor. That's essentially what happens, and the microbes do it. So that community is really important, and when we have problems with too many of those bacteria that produce beta-glucuronidase, we can have an increased risk for things like cancers, right? Particularly breast cancer, colon cancer. And again, if I'm eating a plant-based diet, not a plant-exclusive diet, but that plants make up a significant portion of my diet, I'm getting a lot of fiber. My goal is always 35 grams of fiber. That's going to feed my microbiome, and it actually feeds the bacteria that don't produce beta-glucuronidase, right? So how we eat can influence how many of these little bugs are there, decoupling your estrogen and allowing it to get recirculated, right? This, again, is something we test. We can test it in the stool and we can see, do you have a lot of this? And we can actually control for it. We can help grow some of those good microbes and feed the good ones. And we can also do things to keep the black wrapper around your estrogen and help you get it out. So the last estrogen metabolite. So our gut plays a very significant role. And so beta-glucuronidase literally was one of the things I was looking at. And so I'm one of the world's experts on beta-glucuronidase and what does it do? I'll have to own that because nobody looked at what I looked at, which is its activity in women who are cycling that have IBS and how it has an impact on those receptors inside the intestines. So the reality is it's not really about your estrogen itself, right? It's not really about this. So let me get back. So now that I've explained this kind of complex thing, and I know your heads are probably spinning and you might need to listen to this 2 or 3 times, which I hope you do so you can kind of understand it. So again, I'll go back to the beginning in my working hypothesis. Again, there's nobody that has researched this. This is just me sort of explaining my scientific thought process and reasoning. We have a lot of estrogen and a lot of progesterone and a lot of testosterone when we are cycling, when we are mature, young adults and fertile. And it gets very, very high. Again, your estrogen can get to 4,300 during pregnancy. And so we are protected if we have our period earlier, we have our period longer, and we have more children, right? So if estrogen was the problem, it should be the reverse of that. The woman who was cycling normally, who started early, who had more babies, where she had a higher frequency of estrogen increases, should be at greater risk. And it is 100% the reverse of that. So logic would tell you it is not about the estrogen, particularly estradiol, which is what our ovaries make. That doesn't make sense, right? Because otherwise every 20-year-old would have breast cancer. Now we're seeing it more in younger people, and I'm going to explain why I think we see some of that increase, right? So it's not about the estrogen itself because it's highly protective. When we go through menopause, whatever year that is for you, our risks go up. So now the ovaries, the eggs are empty. There's none left in there. Your estradiol now plummets. Most people are under 5, like almost non-detectable. Mine was almost non-detectable when I tested it before I went on estrogen. So estradiol is nowhere to be found. Your fat cells can make estrone, which is pro-inflammatory, right? And we make some of that. There's always some of it, but it's not a lot compared to estradiol. And we have toxins that look like estrogen, that are not really like estrogen, but they're kind of like that, like BPA, DES, pesticides, herbicides, phthalates, plasticizers. We have all of those, so they look like that. So those are now circulating in your body. And women gain 10 to 15% of their body weight in body fat when they go through menopause. Why? Is it because the body is trying to make more fat to make more estrogen? I think so. And I think it also goes along with a bunch of other metabolic things that I've talked about on this show. But we see fat weight and gain, and we often see higher levels of estrone, which estrone is that hormone byproduct of fat when the ovaries can't make estradiol. And the number of receptors for estrogen in your brain increase as you go through menopause. They don't decrease. So it's so important to the brain. The body's like, I need to capture estrogen no matter how I get it, right? So the body's trying to make estrogen and make it available. The estrobolome, those little bacteria in your gut, that allow you to basically unwrap and take kind of used up funky estrogen-like compounds that you were going to throw out and allow it to recirculate. Why does it exist? Right? Why does it exist? Because if it was all bad, you would think that it would have bred its way out of the environment or something like that. It would kill humans, right? If it was all bad. There is some research out there that indicates that maybe the growth of the estrobolome, which is we see increase in menopause, maybe because the body is preferentially creating an environment for those bacteria to grow, so you can decouple those estrogen compounds that were ready to go to the trash and allow them to recirculate because the estrogen was so important to the body. Despite the fact that it might create some nasty byproduct later on down the road that might grow a cancer cell. Right? Because the reality is if we lived long enough, much like other mammals, we will eventually grow cancer cells because the cell sort of gets a software error as it replicates. It's just what can happen as you age, right? Not in everybody, but statistically it's more likely. So again, your body's like, okay, I'm going to grow a bunch of bacteria that allow me to recirculate broken estrogen because it basically takes it out of the trash and throws it back into the house. Okay, so now we have that. And now we have how your body prepares those estrogens to go out to the trash. And if you don't have the right nutrients like folate and B12, B9 and B12, or you don't have, and you preferentially make a negative byproduct, and the ratio between that first set of liver detox stuff, the red, green, and blue wrapper, compared to the pink wrapper, means you have a bunch of stuff circulating that was ready to go to the trash but couldn't make it. That increases your risk for breast cancer and ovarian cancer and all these other cancers. So my working hypothesis is this, right? And I don't have a way to actually prove this, but I look at that and I go, okay, in light of the fact that we make a ton of hormones when we're younger and we don't see those cancers when we're younger, statistically, not in large percentages, we have a high level of cancer risk comparatively when we are postmenopausal. We have multiple mechanisms in which our body is going through to try and create more estrogen, either by fat cells, either by the microbiome preferentially growing bacteria that actually increase the ability for them to decouple it and take stuff that was slated to go to the trash and allow it to recirculate. And then you have your fat cells making more estrone. I think that the risk for breast cancer is mitigated by hormones when they are the bioidentical hormone that our body makes. Right? Because right now, if you look at the research, the 10 million women study, there was no relationship. There was actually decreased risk when women took hormones and stayed on them. And I've talked about that in many other podcasts. There's a decreased risk for all lung cancer, colon cancer, breast cancer, ovarian cancer, endometrial cancer if you are on hormones and stay on hormones, if you're a menopausal woman. So it's the opposite of what the Women's Health Initiative tried to present. And it's a lot bigger group and a lot larger population and a lot more data. So my working hypothesis is that we have all these toxins in our environment and all these other things that look like estrogen. Those of us who don't get it ready for the trash very well have an impaired way of getting it ready. And that if we don't know that and we don't nutritionally support it, it may be made worse. And that when we lose the estrogen that our ovaries makes, estradiol, which is highly protective, it leaves those receptors all over the body, all over the tissues, wide open for the half-broken BPA, funky whatever plasticizer estrogen that's recirculating, or the 4-hydroxyestrone red-wrapped estrogen metabolite that didn't make it out. It allows that to have a receptor to hold on to because there's no estradiol there to protect it. That's my working hypothesis. Now, with that being said, what things can we look at? So number one, the research prior to the Women's Health Initiative and the research coming out after the Women's Health Initiative, and particularly the recalculation of that data, they have finally agreed and admitted that they were wrong. All is in support of estrogen replacement in women, particularly estradiol replacement in women. And you always replace progesterone with it because progesterone's job is to counterbalance estrogen. It has its own hormonal effect. It has its own hormonal impact, but it definitely keeps estrogen from causing extra blood supply in the uterus and cell changes in the uterus because its job is to do that. And then testosterone. Hey, well, guess what? Testosterone is actually protective against breast cancer, ovarian cancer, and uterine cancer. So we replace testosterone also because that also declines in women. And that if we had those hormones in physiological levels, not where you were at 20 at the peak of ovulation, but at physiological levels, and we check the hormones and make sure they're there, and we also look at how you metabolize them. So this is where urinary metabolites, hormone testing in urine is vital. We can look at it. I can tell you how much red, green, blue, wrappered estrogen you have. Are you making the pink wrapper? I can see those things, and then we can manipulate it. We can help you change your diet. We can help you add supplementation. We can look at that, right? We can even check the stool and see, do you have bugs that are mobilizing estrogen in a way that you don't want to, right? We can check all those things. And I think that's what's really missing in the current kind of blaze the trail of everybody jumping on the hormone train. My team at my clinic has been doing hormone replacement for 20 years. Right? Hormone replacement for 20 years. We didn't just figure this out 2 to 3 years ago and we jumped on the bandwagon when we went through menopause. We've been doing that, and we've been doing hormone metabolism, stool testing, organic acids, genetics since they've been available, right? Because we understand this deeper dive. So I think the next generation, Medicine 3.0, when we're looking at hormones and hormone replacement, requires that level of in-depth look, Because we're also, even if you don't do hormones, even if you don't do any hormones, you could still have things passing through that pathway that are less favorable, that leave you at a greater risk for cancer, right? Even with no hormone replacement on board, your risk is still higher, right? So we could still check that. I think that's where Medicine 3.0 needs to go. That's why I'm so passionate about women's health. That's why I'm so passionate about spreading this information, because yes, there's a significant portion of women that this may not be a concern, right? But I think you can't tell that until you know that. And conventional medicine is so broken in that we don't look for something until it's already present, right? We use, let's say, mammograms to prevent. It doesn't prevent jack. All it does is identify. If anything, it increases risk because we have a lot of radiation. Yes, I think it's important to detect it. And there's other things like QT imaging that is a much better technology. And I can't wait for that to expand across the US. But detection, meaning waiting for a symptom to show so much that you can go detect it, is not prevention. And we have the ability, particularly around these type of cancers, to do something. It may not be everything, but something is better than nothing. And I think for a lot of women, knowing this kind of information will allay your fears around what you know you probably need, which is you probably need hormone replacement because you're tired of the brain fog, the fatigue, the lack of libido, the painful sex, the urinary incontinence, the mood swings. Oh yeah, and the hot flashes and the night sweats, which, increase your risk for stroke and heart attack. I don't know if you know that, but by almost 50%, right? So all of that osteoporosis risk, I think being able to understand that there are things that you can do and look at to identify some of those other things that you think are risks, and that could potentially be risks even if you're not on hormones, and actually hormones may be protective. Because again, I think that if estradiol was on board at low physiological levels that is protective for all those things, those other toxic byproducts wouldn't have anywhere to sit. There would be no keyhole receptors for them. And I believe that's the problem. And I think we oversimplify in medicine because the target was always that we're going to use estrogen receptor as the drug target for delivery. Not necessarily that's the thing that did it itself. It just happened to be the drug target for delivery, and that we're overlooking the biology, the chemistry, and the physiology of the body, right? And until we test that, we don't know. So, of course, I've got my working hypothesis. Don't run around and say, Betty said it, and if this is true, this is my working hypothesis. But I do believe that there's truth here. And I do believe that you can be armed with this kind of data and this kind of information about your own health, so you can make your own decisions, and you can get help and get hormones replaced if that's what you want and if that's what you need. I do believe most women would benefit from that, just from everything I've studied, right? And the, tens of thousands, I don't even know how many tens of thousands of people that we've helped over the years, maybe hundreds of thousands at this point. I know that to be true. So, all right. So what's your take home message? My last little conclusion, right? So estrogen metabolism is complex, right? And I know that you're probably like, that was super heady, Betty. It really was. It's complex. But what you need to remember is that we can manipulate it, and you can check for, do you get your estrogen after you're done with it, to the trash. And the balance between these different stages of how your body preps the estrogen is part of the problem, right? And it's genetically implied, but not 100% controlled by genetics, because you can change your diet, lifestyle, and other things to manipulate it. And regular physical activity, a diet full of fruits and vegetables, nuts and seeds, filled with phytoestrogens and polyphenols, can help estrogen metabolism. So you got to have those foods in there, and it's not, veggie sticks that look like French fries that are coming from a bag that are really kind of, I don't know what they are. Don't fall for that. It needs to actually be in its original form. And the microbiome actually plays a pretty significant role in estrogen metabolism and hundreds of other things too, right? Just because we haven't studied it doesn't mean it doesn't exist. And this knowledge really is power. And by understanding this, you can make informed decisions, and you can understand your health better. And so obviously, if you need help, reach out to us. We'd like to help you understand that. That's why I built my clinic. That's why I built Minerva, our telemedicine company, because I wanted to be able to give this to women across the United States. I didn't want to wait until the medical system 30 to 40 years from now tried to figure some of it out, right? The average is 18 years for clinical research to make it to clinical practice. This would probably take longer just because it's more complex. All right. I hope this was helpful to you. Until next time, this is Dr. Betty. reminding you to have a powerful and awesome menopause journey. Stay curious, stay healthy, and keep mastering this lovely game called menopause.